see everyone. It's my great pleasure to introduce Niko Hildebrandt, our newest, probably newest, professor in the Department of Engineering Physics at Luminescent. He recently started. He holds a diploma in medical physics engineering and a PhD in physical chemistry. Previously, he was a research group leader in various institutes in chemistry, physics, and biology, situated in Germany, Germany and France. He was made a professor at University Paris Saclay from 2010 to 2022, then at Luminescent from 2022 to 2023, and was recently at Seoul National University from 2021 to 2023. His main research interest is time-resolved photoluminescent spectroscopy and microscopy, and as well as the application of lanthanide and nanomaterials for multiplex force-through resonance energy transfer in terms of biosensing and imaging. So without further ado, I will let you take the floor, and we're super happy to hear you talk today.
Thank you very much, Catherine. Or I should say Catherine, no, because that's the original. I run around so that Florian always has to. I'm sorry if I do that, but I know you're good in following the speaker. Well, thank you for the invitation. I'm also sorry for all the people who thought, the newest member should be a young person, and now you got an old guy without hair talking to you, but I hope I can still make it a little interesting. I will talk about FRET, which is written already here. There's a picture of Theodor Förster, who's the, we say inventor of FRET, and then a bit of biosensing, and I'll also introduce myself a little more as I'm new here. It just worked before. I got to be closer. Wow. Oh yeah, it does work now. I was born in Germany, so between Hamburg and Lübeck in the northern part of Germany. So that's Denmark. It's almost Denmark, and lived there in a village called Bad Segeberg. It's much cooler in English because it's Bad Segeberg, and it sounds really vicious, but it just means bath. You can see our lake. So I lived on the other side of the lake. It looks like a rocket from here. Then I moved to Berlin for studying at the Berliner Hochschule für Technik. At my time, it was called differently, but in Europe, universities like to change their names and their logos all the time. And I think in between, it was called University of Applied Sciences when I was there. Then they changed it to Beuth Hochschule, but then they found out that Beuth was unfortunately a Nazi, and they hadn't found that out, so they changed it back to Berliner Hochschule. This is the campus here. Then I did my PhD in Potsdam, which is close to Berlin, at the campus of Potsdam in Golm. Then moved over to Paris, close to Paris, actually closer to Versailles here, where the chateau is. This is obviously Paris. This is the campus in Orsay, also very green. You might think there's no building at all, but there are several ones. Then I moved to Rouen, which is here in the Normandy. Also very nice place, very old city. The campus is not so nice, but the city of Rouen is quite nice. So this I did until very recently. In the last 2 years, I had some Asian adventures. Okay. Are they still on the? Now it works? Not on Zoom. And let's see how we do that. Because here it's on the third one, right? Well, if I swap here, now you see the wrong stuff, right? Well, I unshare and then share again, right? New share. Which one do I have to share now? That one, maybe? Let's try. What do you see? You see the wrong. So now I need to switch up here, right? How about that? Okay. Switch. It even switches with that. Okay. Good on Zoom? Okay. I'm sorry for that. Technical difficulties as usual. I had some adventures in Asia as well. Recently, I was in Seoul in South Korea in the Department of Chemistry. Seoul is a really beautiful city if you like big cities and if you like hiking and mix of technology and culture. I was there with something that's called the Brain Pool program. If you're interested in ever going to Korea for a year or two, let me know. They made me an ambassador of this program, so I'll be happy to tell you everything about it. I expected I bring a brain, they give me a pool, but it doesn't work like this. It's just their names, so you have to do actual research there somewhere. And that's where I spent my last 2 years in the group of Jwamin Nam, who's doing nanoplasmonics. I think I don't have to, always when I go there, I don't have to introduce that slide at McMaster, but well, you know where it is. So I changed my introductory slide because a few days ago, quantum dots, what we work with, got the Nobel Prize. So I thought it's nice. I only need a single slide for introduction now. And even when I talk to people, usually they ask me, what are you doing? I said, well, we're doing biosensing with fluorescence. They said, oh, no, wait, we're working with quantum dots. Now everybody knows what it is because it got a Nobel Prize, these 3 guys here. If you look up there, this was a conference in Paris, which I attended, where actually all the 3 of them were. You can see this is Louis Bruce here, this is Manjiba Wendy, and this is Alexei Akimov. And so there you can learn, if you're at a conference, you should always look into the camera, because I am behind that guy up there. You see this piece of skin, that is me. And I'm sure that it's me because this is one of my students, and this is Nicola, a colleague, and he's talking in that direction, and another one. So it's me. So I was there. You have to believe me. Our other contribution to quantum dots was this cover. No, it was a table of contents image on one of the reviews that we had about quantum dots. So I thought I look into the Nobel Prize a bit more, and then actually thought I must be really old, because all of the things that are mentioned in this Nobel Prize, we've worked with those. So RNA and laser pulses. And I thought I should also integrate literature. For sure, I didn't find a lot, but I found an interview where Jon Foss actually said, when he talks about, this interview is actually quite nice, where he talks about his life, and with 7 years old, he almost died. And then he tells when he went out of his village, and he said it was a very shimmering and very peaceful, a very happy state, like a cloud of particles of light. So that even fitted in the Nobel Prize of today. I had to look a bit longer, but I also found an interview of Narges Mohammadi, where she hopes that one day light takes over darkness and the Son of Freedom embraces the Iranian people. So that was recently published in the New York Times, also worth reading this. So if you mix that all up, then what we get is basically FRET, and I'll explain you more what that is now. So FRET is an energy transfer between a donor. It needs to be a fluorescent donor molecule or nanoparticle, and an acceptor. What I like of FRET is, first of all, that it's been cool, because I don't know if you know David Bailey. It's a photographer, and I was in London in the 90s, I think, and I bought this poster of Michael Caine. Some of you still know Michael Caine, I guess, so that's him. He's still around, so he's playing now always in the Batman movie, so that's where the young people know him from. And this poster has always been, since I have it in my kitchen. So no matter where I traveled, it will also be, now it's still enrolled, but we move to Toronto tomorrow, and then we'll put it up the first thing in the kitchen. And this is the picture of Theodore Förster from 44. While David Bailey hasn't made a photo of him, I put it inside there, but I think it's also quite cool at the 40s, where as a scientist you could still smoke in the lab, and while you're making experiments. So, and obviously I'm not saying that smoking is cool, it's just because he's got the cigarette as well there. Well, otherwise, Förster resonance energy transfer is interesting because we can make this energy transfer from a fluorescent donor to an acceptor if their energies overlap. So the fluorescence of the donor needs to overlap with the absorption of the acceptor. And if they're in close distance, so up to about 10 nanometers, this energy transfer works to dipole-dipole interaction. You can see here that 50%, that's what we call the Förster distance, is usually below 6 nanometers. That means, so let's say it's 6 nanometers here. That means at half R0, at 3 nanometers, we're at 100%. And at 1.5, so that would be 9 nanometers, we're at almost 0%. That means we have an extremely high resolution, which is angstrom resolution or sub-nanometer resolution between this donor and the acceptor, that we can measure with these interactions, so with measuring spectra. And it's actually quite easy. So I think my battery is running out here. But on the bottom right, you see that the luminescence detection with intensities. So we always need 2 experiments. We need to measure the donor in the presence of the acceptor and the donor alone. And once you have those, you take the intensity, divide the donor in the presence of the acceptor by the donor alone, and then you have the FRET efficiency. And with this equation a little higher there, you can see that we can measure the distances. So this R naught or R0 is what we call the first distance. So that's 50% efficient. And this R would be the distance between the donor and the acceptor. And thereby we can measure the donor acceptor distances. Classic FRET pairs are dye-to-dye FRET pairs. So for example, fluorescein is shown here, and rhodamine. You can make energy transfer between fluorescein and rhodamine. So you can excite the fluorescein, as you can see here on the left side, that's the absorption spectrum. The red one, you excite it there, then it will weather emit, which is the blue spectrum, or transfer its energy to the acceptor, to the rhodamine, because it overlaps with the emission of the fluorescein, and then you can get emission from the rhodamine. Another classic example, which is now classic because everybody knows fluorescent proteins, is CFP, YFP, which are the two mainly used fluorescent proteins for first resonance energy transfer. The nice thing is you can express those guys in the cell. So the cell synthesizes the fluorophores for you, and then you can make energy transfer between those and see, for example, here you have a sensor domain and a ligand domain. And when there's something that you want to sense, it will close or open, and this difference in the intensity of the donor at, for example, 475 nanometer and 530 nanometer gives you a quantitation or a quantitative value of the concentration of the marker that you want to measure. In our lab, what we've been doing and what we will most probably continue to do here, among other things, we're using lanthanide complexes as donors and then make energy transfer to fluorescent proteins or to dyes or to quantum dots. And then when I did my research, I found that this was actually the only missing Nobel in this diagram. So for dyes, already in 1905, Adolf von Baeyer got a Nobel Prize, then Jean-Marie Lehn and others got the Nobel Prize in '87 for supramolecular chemistry for these complexes. And the fluorescent proteins got the Nobel in 2008. We're also adding, so usually my slide looks like this, but there's no Nobel Prize for upconversion nanoparticles. We also use nanoparticles as donors. These upconversion nanoparticles that also exist of lanthanides that can make energy transfer to these different acceptors or donors. So in that case, where there's two arrows, we make a first energy transfer to the quantum dot, and then the quantum dot will make a second energy transfer to a dye or to a fluorescent protein. So these lanthanide complexes are quite special because you need to protect your lanthanide. Lanthanides in solution, so if you take terbium chloride, for example, and you put it in water, the extinction coefficient that you can see here on the left side would be about 1 or maybe 0.5 per molar per centimeter, because they're efficiently quenched by water. So if you put these ligands around here, and you can see here 3 examples of such complexes, 2 with terbium, 1 with europium. These ligands, first of all, coordinate completely the terbium or the europium ion. That means that almost no more, so in this case, there's still 1 water that can coordinate. In this case, there's 9 coordinations. There's no more water that can access the coordination sphere. The terbium is not quenched by water, so it will have a high quantum yield. Moreover, these complexes here are like light antennas. They collect the light from outside and then transfer it to the terbium. So there's already first energy transfer step within these complexes, and thereby you can increase the extinction coefficient by several 10,000. So you can see here it's about 25,000 compared to 1 is a little higher. The disadvantages may be that we have to excite them in the UV, but the nice thing is that between excitation here at 350 nanometer and the first emission line at 500, this is very easy to separate by using filters or mirrors. And then we have these emission lines here, and if you don't know lanthanides, you might think these are 1, 2, 3, 4, 5, 6 different fluorophores, but these emission bands are all from the same terbium. So there are many different energy levels. I think terbium has 3003 energy levels, and some of them can emit light. And this is the lowest energy, the highest energy, and this is the lowest energy one. And they also have, apart from these very narrow and separated lines, a very long emission lifetime. So you can see the fluorescence lifetime on the bottom right is milliseconds, whereas quantum dots or dyes of fluorescent proteins would stay in the excited state in the average for a nanosecond. So this is 1,000,000 times longer in the excited state. And a millisecond is extremely long for anything that is fluorescence. The other lanthanide we're using are upconversion nanoparticles, and they're interesting because you can convert 2 low energy photons into 1 high energy photon. I'm saying 2, but you can also use it with 3 or 4. So multiple low energy photons, for example, red, as you can see on the bottom left, can be transferred into blue. And this is not like 2-photon excitation where you have femtosecond lasers with high energy. So you really excite, you need this high energy, this high pulse energy and femtoseconds to excite the molecule at the same time. Here we don't need it because the lanthanides are the slow guys. So we excite the first real state. So this in-between state on the bottom left is an excited state. You excite that first, it stays there for at least a few microseconds. So you have enough time to excite it again in the second excited state. And from there, you can de-excite. As you can see in the middle, there's many energy transfer pathways. And then you can get from the 2 red emissions, you can get out blue, green, or also red emission. On the right side, you can see examples of erbium or thulium, for example. So here, the advantage is we can excite in the NIR. And in the NIR, you don't have almost any autofluorescence from your cells or from your biological samples. So you excite in the NIR, but you can still use your normal microscopes because the emission will be in the visible. So that is an advantage that people like upconversion nanoparticles. Some people call it deep tissue imaging, but if you don't know anything about deep tissue imaging, you will think, well, deep tissue imaging means like X-ray. You can really go deep in tissue, but deep tissue imaging in that case means maybe a millimeter instead of a few micrometers. But it's often enough to get through, and you can do that experiment yourself. So, I mean, my laser pointer is not working anymore, but if you use a green laser pointer, you can't see anything. But if you use the red laser pointer that you have, and that's what they use in the hospital for measuring the oxygen, you can see that it goes through because red light penetrates deeper into tissue. But for sure, we have fluorescence, which is much weaker, so we can't go through our fingers. We also work with dyes. Dyes we basically buy off the shelf. You can get them from the blue to now the NIR, and you can get them in different brightnesses. You can also synthesize them yourself, or you ask biomolecules to do it, proteins. So here are fluorescent proteins, and they use the same dyes. So if you look on the top left there, these dyes have a little bit the same structure as the organically synthesized dyes, but it's the protein that synthesizes them, and they are in this barrel, in the center of the barrel of the protein. You have these dyes that are doing the emission of the fluorescent proteins. And the great thing is they come now in all kinds of colors that you can also buy, and you can see this is already from 2009, so you have even more colors now, and you can express them directly in the animal that you are investigating or in the cells, or you could even do it in yourself if you like. Quantum dots are great because you can tune the color with their size and their material. So in the middle, you can see cadmium selenide zinc sulfide core shell quantum dots. And if you make them small, so with a diameter of about 2 to 3 nanometers, they'll be blue to green. So they emit at 510 nanometers. And if you make them larger, they shift to the red. So basically, you start your synthesis, and if you stop earlier, they're blue. And if you stop later, they're red. But this is just the simple. I'm not a synthetic chemist, so this was how it was done before. Now the people who do it have much more sophisticated methods, but by the size you can tune them. And if you look on the left side, you can also see if you change, for example, from Cad sulfide to lead selenide, you can span, or you can go from almost the UV, so from up 400 nanometers into the NIR to make these different quantum dots that have a very high extinction coefficient, so they absorb much more light than dyes, and they have these tunable and also very narrow emission bands. So with a dye, you have usually a first band and then a shoulder, and here you have almost Gaussian profiles, or well, some people prefer it maybe Lorentzian profiles. I don't know, but it's at least very symmetric profiles of the emission that you have. So if we put these lanthanides and dyes or lanthanides and quantum dots together, we need the spectral overlap, as I told you before. So the emission of the terbium that you can see here in black needs to overlap with the absorption of the dyes on the left side or with the quantum dots on the right side. And this overlap determines the first distance. So the distance where FRET is 50% efficient. And you can see I took 5 dyes off the shelf here. They all overlap more or less with the terbium, and we can get first distances in the range of 4 to 6 nanometers, which is already nice. But with quantum dots, because they have these extremely high extinction coefficients, so if you look at the y-axis, it's 10 times higher for the quantum dots than for the dyes. And also the quantum dots overlap almost completely with the emission of the terbium, which means we have a very large spectral overlap, and then we can get first distances of up to 11 nanometers. And if you go to the textbooks, you will read FRET works in a distance range of 1 to 10 nanometers. So with these systems, we have 50% efficiency at 11 nanometers, which means we can extend the range of FRET using quantum dots. And when I'm saying this, some of you might have already thought, well, it's partly a lie because the quantum dots have a size on their own. So a quantum dot with 10 nanometer in size, if we're using 10 nanometers, and we're measuring this distance from the center of the quantum dot, so we have to subtract 5 nanometers from this actually, because first we cannot get the molecules into the quantum dot. So the message here is we can have large first distances, and we can transfer energy from one terbium to different dyes or different quantum dots at the same time. So now if we measure the donor, the terbium, and we see the quenching, we don't know which acceptor quenched the terbium. So we need to look at what we call the sensitized emission. So sensitized in that case means that the acceptor receives the energy from the donor. We use that word all the time. Some years ago, we wrote a review article about FRET in Nature Methods, and the editor said, well, you cannot use this word sensitized. It doesn't exist. And if you ever wrote a review with some Nature journals, I think that's how they do it. You write the paper, but then the editor rewrites it. And you say, well, why don't you become the author and tell us what to write inside? You said, well, sensitized doesn't exist. We said, okay, we called it different. I can't even remember how we called it, but I think sensitized emission makes sense. So it gets sensitized from the donor. And then the emission that you see here are the emissions from the quantum dots or the dyes. And these we can distinguish now. So we know was the energy transfer from terbium to blue dye or to blue quantum dot or to the brown or orange quantum dot. And if we use the quantum dots and use these filters, these are the shaded lines in the background, then we can very well distinguish the different quantum dots and the different energy transfers here. These bioassays that we're using them for have existed since a long time. They're called homogeneous or rapid solution phase assays, where we have 2 antibodies, and these antibodies are both specific for one biomarker. It might be a cancer biomarker or whatever else. And if they're free in solution, as you can see here, you excite them in the UV, then you get a very long emission of the terbium, and you get a very short emission. So this is this orange thin line just in the beginning of time there of the acceptor, the dye, or the quantum dot, and you get a lot of autofluorescence background from your sample. So now if you add a drop of blood with this biomarker inside, then the antibodies will bind, and now the terbium and the dye on these antibodies are close together within a FRET distance, and from there we can make energy transfer. So that means the terbium is quenched now. So the terbium, which had a lifetime of, say, 2 milliseconds before, it is quenched to, let's say, half. So it's 1 millisecond in lifetime. But what is more important, that the very short lifetime of the acceptor, which was nanoseconds, now also becomes millisecond. Because the sensitized emission, so now the energy doesn't come from the light, but the energy comes from the excited slow state of the terbium, will adapt the same lifetime as the donor, plus its own lifetime. But now 1 millisecond plus 1 nanosecond is still very close to a millisecond, so you don't see it. And all this orange signal that you see here only occurs if you have the biomarker, this antigen, to detect. And now we used pulse excitation and time gating, so we measure the intensity only in this time gate with our detectors, and that is free of any autofluorescence and free of any background from direct excitation of the acceptor. So we can measure very sensitively without separation. And that is a big advantage in biosensing, because often you have to incubate your cells, you have to wash, and all these washing steps have been gone. Or if you take an ELISA assay, you have to put first the antibodies on a solid phase, then you have to wash, then you incubate with your biomarkers, you have to wash again, then comes the second antibody and you have to wash again, and that takes time. And here you just mix and measure, because all the other signals will be different from the single antibodies. These things can be done on plate readers. So on the bottom here, you can see commercial plate readers. There are probably more. We also work with a clinical plate reader. This is called Cryptor from Thermo Fisher. They're mainly into sepsis and cancer biomarkers and cardio biomarkers. We also develop our own readers and our own microscope to make these time-gated things, but it's nothing very special. So the people in drug screening, they all know these systems, which are called HTRF, and this is nothing else than time-resolved FRET. Some also call it T-FRET or TR-FRET, I think. So the advantage is we can make color multiplexing. That's what I showed you before. So one terbium and different dyes. So we can, for example, measure 3 different DNAs, as you can see here. Well, you see 3 same DNAs, but it's just because I was too lazy to draw 3 different DNAs, but you can imagine the principle. The other thing we can do is time multiplexing, because with this FRET, I hope you understood that the quenching depends on the distance, right? If we're closer, the quenching is stronger. That means that the lifetime that was before like this becomes shorter. And if the distance is longer, the lifetime stays. So if we now can make systems where we can tune our distance, and DNA is great because each base you add adds 0.33 or 0.34 nanometers. So we can tune that distance. And with these different distances, we get different decays. And on the bottom here, you see, I would say it's maybe a 3 exponential decay. And the different colors are the different concentrations. So we could measure 3 different DNAs of different sizes here, because they will have a different donor acceptor distance. And we only need 1 pair for this. So we don't need 2 dyes, but we use the same terbium and the same dye acceptor. And we call that time or temporal multiplexing. And if we can combine those together, time and color, we can make what we call spectrotemporal multiplexing. And if, for example, you combine 5 colors with 3 time gates, you can get in solution 15 different biomarkers. And if you can spatially separate it, so for example, on some fixed products or in imaging, it's not 5 multiplied by 3, but 5 to the power of 3. So you will have 125 different codes. And if you think that further, and that works very nicely on a PowerPoint slide, not so much in the lab, but if you take 5 colors, 3 distances, and 3 intensities, that will be 3 to the power of 3 to the power of 5. That could give you 14 million different codes that you can create with that. So theoretically, that is not difficult, but if you want to do it in reality, it's a bit more difficult. So what can you do with this? And I want to give you some applications we've performed over the years, mainly based on quantum dots. I'll be happy to talk also about all the others. And well, as you know, as I'm the new young member of the faculty, I'll be here at the moment. I have not a lot of committees and teaching to do, which doesn't mean I don't have anything to do, but my schedule is a bit more free. So if you want to talk and chat about something, you have a good idea, just let me know, drop me an email, and I'll probably pass by within a few minutes if I don't get lost on the campus. So in quantum dots and diagnostics, the size matters because the quantum dots have around the same size as an antibody. So here is a classic quantum dot, about 10 nanometers. This would be a core shell quantum dot, maybe with a coating. They can have a diameter of up to 10 nanometers. A full IgG antibody, immunoglobulin G, as you can see on the top, has about 150 kilodaltons. That's also about 10 nanometers. It's even a bit larger from top to top. It would be maybe 12. And if we reduce it, so we cut off the FC region on the bottom, we get FAB2s. We can cut it more, then we get FAB. If you have llamas or sharks at home, you can make nanobodies. You can also synthesize those, but the llamas and sharks express them. Or you have these artificial antibodies that are derived from proteins. We work with ADAPTs or alpha reps. You can see there's a big size difference from about a factor of 20 going from an ADAPT to an IgG. And they all have the same function. They're binding, like the antibody up there, to some specific marker. And so by doing this, we can tune our distances and get closer to have higher FRET efficiencies to make more efficient sensing. We can also orient the antibodies. So the other way is not to put it like that on the quantum dot, but to orient it like that. And as the binding site is on the top there, you can also get closer. So there's a lot of different surface modifications and bioconjugations which we need to do to optimize these markers before we can, these sensors or these probes, before we can do any actual biosensing. So one example which we did with quantum dots, and this is a terbium to quantum dot energy transfer, is with nanobodies. So you can see here three different quantum dots. Well, it's actually two different quantum dots. It's one emitting at 625. And the CL4 means that this is a cadmium selenide quantum dot, which only has a very thin coating of chemical ligands on it. So that's maybe a nanometer thick, but it's still enough, if it's well done, to protect the quantum dot from water and to work into solution. And then we have these nanobodies, so I call them NB, this is NB2, and the top ones have a his tag on it. So this is a tag, it's a histidine, 6 histidine tags that is often used for purifying your proteins. So when your cells produce proteins, you want to purify them, you have a column, and they will stick on this column, these 6 his tags. And that is good. We have them for the nanobodies as well. And our quantum dots are like a column because they have zinc on the top because it's a zinc surface, zinc sulfide shell surface. So they will self-assemble to these quantum dots. So we don't have to do anything but mixing the quantum dots with the nanobodies, and they will go there if they have a his tag. And then we have the other nanobody with a terbium, so that's terbium nanobody 1. And if we add the epidermal growth factor receptor, EGFR, they will form these sandwich complexes like I showed you before with the big antibodies, and then we have an energy transfer. We also did it with 2 other tags, so biotin tag or cysteine tag, that also works. And you can see these are always the kinds of curves that we're measuring in our lab. We have a concentration of the biomarker. This is the epidermal growth factor receptor in our case. And then we have a FRET signal. This is the relative FRET ratio. This is a photoluminescence ratio of the acceptor divided by the donor. And we can see that it increases. So by these curves, we can see at which intensity of the signal we have how much epidermal growth factor receptor. So for the green curve, for example, we measure FRET ratio of 5. We know in our sample we have 5 nanomolar of the epidermal growth factor receptor. This is nice, but we still need 2 antibodies as usual because we want these sandwich immunoassays. But it would be great if we could do it only with 1 antibody or nanobody. And so I would say we developed the system, but this system developed itself because the student made an error in the lab by mixing things together, and then some things were displaced. So it's often good to explore things. Let's say, well, I shouldn't say she made an error. Also, it was a very nice study afterwards. So she explored her system nicer and put these nanobodies to the quantum dot, which I said they don't make sense, which also means you shouldn't always listen to your boss, but think, yeah, you know, we only think on PowerPoint slides, right? So as a student or postdoc, you think more in the lab. So add whatever you can to a quantum dot. It might give good results. So here she added these nanobodies with a his tag to the quantum dots, and they were taken off by the EGFR. Or they were taken off and we didn't know why. And then we tried to put the terbium nanobody itself on it. So it went on the quantum dot. This is what we didn't want because we wanted them to be independent. So this his tag terbium, and once we add the EGFR, it displaces it. So that means that our signal, as you can see on the bottom left, decreases because we get less FRET with increasing concentrations. But for sure, we can use also a decreasing ratio to measure our concentrations, and we could see here that works quite nicely with only 1 nanobody with a terbium. The quantum dots we can use alone, which is great for storage because storing a quantum dot with antibodies on it is a bit more problematic, and we also need less reagents and have a very simple preparation for this. Another antibody we used, or this artificial antibody, are these alpha reps. These are artificial proteins that are composed of these heat-like repeats. So they're always repeating. They have this repeat-like structure. And in this case, we had two. So on the bottom, it's the easiest to understand. So we can connect them. So we have the two orange ones, or an orange and the blue one. And they are now connected on the quantum dot. You can see one has a his tag to the quantum dot. And on the other side, we have a single cysteine where we can put our terbium. And now we have an energy transfer when there's no biomarker. But when we add the biomarker, you can see we can add two blue, or one blue, or a green, or one blue and the green. And they both bind to, well, the blue binds to the orange, and the green binds to the blue. So the other is cyan. So if I said twice blue, well, you see it on the bottom, right? So that you can have an opening, and the opening increases the distance. And this means we also don't need a sandwich of formation, but we have everything on the quantum dot already. And by opening once or twice, we can measure two biomarkers, one after the other. And the nice thing is, as FRET also works for measuring distances, as I told you before, we can measure these lifetime decay curves. And as it's depicted on the left scheme there, we can then measure conformational changes of these proteins on the quantum dot. So how opens it? How does it switch? How does it go over? Because we have now a resolution of maybe 0.9, 0.8 nanometer that we can follow in solution. And the nice thing is there's no crystal structure here. We don't need to put it on any grid. We just measure it where it is, in solution, in the cells, because we only need the fluorescence from it and can measure these structures under physiological conditions. Second example is going to DNA. So first was proteins and antibodies, and we work a lot also with DNA or RNA to develop these multiplex sensors. So that technology, we had a good brand name for it. We called it FRETman, better than Tatman, stronger than Batman. And but that's as far as it went, not further. But, maybe one day I'll commercialize it and I still keep it. So the first idea we had was to, well, again, take terbium, the quantum dot energy transfer, and measure microRNA, because microRNAs can be important tumor markers or markers for other diseases. This is also a discussion. At some point, microRNAs have been a very hot topic because everybody thought they are very important as biomarkers. But then there's so many studies where different microRNAs are giving different signals for anything that it's going up and down. It's maybe like with quantum dots. Quantum dots were almost dead until 2 days ago when it got the Nobel Prize, so now everybody loves quantum dots again. So the idea here is what we want to measure are the mirrors, the microRNAs. We want to measure 3. So we put a small capture DNA on the quantum dot and a reporter DNA. We put our terbium, and these parts are can hybridize, so they are 100% complementary. You can see that on the top there, the red part and the red part, but it's only base pairing of 6 bases or 7. So that means that it's not very stable. It goes on and off, and it depends on the temperature. But once we add the microRNA to it, we have a combination of base pairing, but also base stacking. So that means when we have the microRNA, I think this is the only animation I have in my slides, then they can come together and form these stable complexes for energy transfer, and then we can make energy transfer from our terbium to the 3 different quantum dots. Then these curves get a bit more messy. You know, we have different samples, and the pointed lines are the concentrations that we know, and the others are the concentrations that we measure from this triplex assay. We can find them quite nicely. And what people like me like to do, we then put it in mole and not in molar, because pico sounds smaller, right? But actually, if 0.3 to 1.5 picomole, because we have small volumes, means 2 to 10 nanomolar. This was a nice study from the nano point of view, but if you go to a clinician and say, wait, we can measure you microRNAs at 2 to 10 nanomolar at 3 different colors, they said, yeah, I give you 3 dead patients, maybe they have enough microRNA, but it's not good for measuring that as an early biomarker, because we want to detect cancer early. So you need to go to at least picomolar, maybe atto or femto or attomolar. So that is one thing. The other thing is you also want to go to real samples, measure it in tissues, measure it in blood, measure it in cells, because no clinician is interested that you can measure it in your buffer composition in your lab. But you need to start with something, right? So we said we have to sacrifice our simplicity by adding an amplification. We never wanted to do that before. We wanted to do easy assays, but we said, well, for this case, we have to go down or up in sensitivity, down in detection limits. So we took the system, we didn't develop it. It's called rolling circle amplification by target recognition of this padlock. So you can see on the top, you have a padlock DNA that's a linear DNA, and the ends of it are complementary to the target. So that means if you have a target, it closes like a padlock over it, and then you have to close this nick over the splint. So you can see that in the second one, you use a ligase, you ligate it, and what you have now is a circular DNA. So the target plus the ligase give you a circular DNA, and you have this small target on it. And this is a primer for a polymerase. If you add a polymerase, and you also need to add the nucleotides, will start to synthesize new DNA around this circle. So it will start on the 3' end on the right side of the blue and start synthesizing it around once. And then it arrives at the beginning. But if you use, I always call it a stupid polymerase, it will not know that it has done it. It will just continue and continue and continue because it always wants to go forward, and it goes around like 1,000 times, which means it synthesizes you 1,000 times the same strand that you can see on the bottom there. At one moment, we stop the rolling circle amplification, and then we have 1,000 times a sequence that we know because we designed this padlock. And on there, we can add our FRET pairs now. So we take small DNA probes, add them on there. And for sure, we can design, as you see on the bottom, on the top right, we can use different colors. And we can also use different distances because we know the DNA sequence. And now we can combine here 2 colors and 2 distances, which would give us 4 different DNAs or RNAs at the same time. The limits of detection are great, so we can go down. So now we can use zepto, 40 zeptomole. 40 zeptomole sounds almost like nothing, but if you think that it's still 2,400 microRNAs, that sounds a lot. So you only put that on the slide and not on the paper. You only put 40 zeptomole, which is 300 attomolar, which is still quite low, but PCR can, or they say they can measure single copies of DNA. Although most of them can probably do it, but you can't distinguish it. So 25 is still quite okay. Now this gets even messier because we can measure 4 targets, but as you can see here, we can measure 4 targets at the same time at lower picomolar or sub-picomolar concentrations, which was good enough for measuring microRNAs in plasma. So on the top, you see plasma from ovarian cancer samples and healthy control samples. You can see that RCA FRET could distinguish, so that's the red, and RT-qPCR could not very well distinguish the different samples. For tissue, it's a bit easier. You have higher concentrations, so both of them could distinguish. And we could also do this in cells. And we also found that we always have that RT, so reverse transcription qPCR, always has a signal that is 3 times higher than FRET. And we found that this is due to non-specific signals that you have in RT qPCR. And we had to do a lot of studies because no reviewer believed us that we're more specific with this than qPCR, because they say we always use reverse transcription qPCR. And Thermo Fisher tells us it's great. So it must be great, right? Because it's shown on TV. Or you don't say that anymore. It's because it's shown on, I don't know, because Instagram says it, then it must be right. But we showed that actually it's not extremely specific. We also could use the same technology to measure point mutations. Here we use the BRAF gene for it, and we can measure the wild type and the mutated at the same time because we can do multiplexing. And we can also turn it around. So now we don't want to measure the DNA, but we want to measure the fuel. So usually we have to put a polymerase and the nucleotides inside, but sometimes in cells, for example, for drug discovery, it's important to know the concentrations of nucleosides in the cells. And so we can do this by measuring. So if we have 1, we always add 3, and we leave 1 out. The rolling circle amplification will stop at one moment because there's no more fuel. We do that 4 times, and then we can analyze it, and we use that for measuring some drug studies, as shown on the right side. Then we thought, why don't we transfer that to quantum dots? And this is, I think, the nicest table of contents image that I drew myself ever, and it was also accepted. Because, and that was a really nice project because it's a paper that is full of failures. It's actually only failures because that student started and wanted to do this rolling circle amplification with quantum dots. And that was her main project that she did over 3 or 4 years. Luckily, she published some other papers along the way. But if you think about it, if we combine FRET, which is small on the, it's like a 5 nanometers, quantum dots that can go up to 25 nanometers with coating, and this amplification that you see, that these are actually quite big blobs up to 250 nanometers, and you mix it, it will not just give you the QD FRET DNA fly catlifant at once. On the PowerPoint slide, yes. But so she made, I think, 6 different approaches, and on the end, she found one approach that worked, but which had the same sensitivity as with the dyes. So it was still picomolar, but it wasn't a success. But the story is still nice, and I think it's also my largest supporting information file of, I don't know, 80 pages or something like this. But I like that study quite a bit. So for the last 5 minutes, do I have still 5 minutes? The last 5 minutes, I want to show you how we can use these systems for imaging. And in that case, we imaged the development of zebrafish embryos. So the embryonic development of a zebrafish is shown here. You have one cell, and then the cells divide, and you have many cells, and it goes quite quickly, as you can see here. So from 0.2 hours to 72 hours, you have quite a development stage. And what we wanted to show is if we inject our quantum dots once at the cell 1 stage, how long can we see them? Because you have a cell division and another division, so you go from 1 cell to thousands of cells. Are they bright enough? And can we actually use our time-gated FRET, so from terbium to quantum dot, to get rid of all the autofluorescence? Because I told you before, autofluorescence is not there, but if we do it in our lab, the students often ask me, yeah, but there is no autofluorescence in our samples. I say, yeah, because we use buffers maybe, but if we go to blood, to cells, there's much more autofluorescence. If we go to living animals, there's even more autofluorescence. And so we made a study on these poor zebrafish larvae. So the system you can see here, it's a quantum dot. We have again a peptide. You can see these 6 dots. That's a his tag. So now the terbium with the peptide is attached to the quantum dot. We also add some maltose binding protein. That's the green protein on the top left. That is just nice to microinject because you can microinject quantum dots easier if you put some maltose binding protein on it. Don't ask me why. For some quantum dots, it works with, for some without. For these quantum dots, maltose binding protein was great for microinjection. And if we express fluorescent proteins in the cells, and they also have a his tag, then once you have the quantum dot in the cell and the protein expresses, sorry, the cell expresses the protein, then it can self-assemble in the cell to the quantum dot because of the his tag that it has. That's what you see schematically on the left side there. And then we can do energy transfer from terbium to different quantum dots. So if we use different colors, and also from terbium to this M-cherry protein, or also from a quantum dot to an M-cherry protein. So you can see that here, terbium transfers to turquoise, orange, red quantum dot. It also transfers to the M-cherry, but the turquoise quantum dot also transfers to the M-cherry. And the principle is shown on the right. You have the excitation at time 0. The background decays until about 10 microseconds. And then we have this background-free time-gated detection of these 4 different color channels to distinguish between different cells, for example. So if we microinject, you can see here we first worked in live cells. The middle one is the case where we injected all of them at the same time. So the terbium turquoise, terbium orange, terbium red, and you can see energy transfer from all of them. So you can see the donor and the 3 acceptors. And on the bottom is there we see the attachment of the fluorescent protein. So you can see that we have a few cells. So the one which says time-gated FRET with a cyan, you can see there's some signal on the right side which is not there for the magenta. That means just that in this cell, the M-cherry wasn't expressed. So your cells don't always express, all cells don't express. And if you're lucky, you find one field of view where you have some that express and some don't. I say lucky because then we have the control experiment directly there. So you can see that we have terbium and quantum dots both in there. The terbium signal is a bit less because of the energy transfer, but there's no energy transfer to the fluorescent protein. But the cells on the left side that contain the fluorescent protein, there you have these three-step energy transfers. You can actually make sensing inside the cell, recognition of this M-cherry to the quantum dot. Then we injected them in the cells, and so here we inject them at time 0 in the 1 cell stage, and there you see 4 hours, 24 hours, 72 hours, and you can see that in the CW, so this is constant steady state excitation. In green, you cannot distinguish the background from the quantum dot signal because they're both very green. In red, it's a bit better, but you still have a lot of background. And the middle images show on the right side. So on the left, it's time-gated FRET with terbium to quantum dot. You see signals, and you have no autofluorescence background at all, which means it's all signal. We can measure those over several days. So there's no second injection. There's one injection only. And after 168 hours, that's what, 7 days? Yeah, I think after 7 days, you can still see, you see it gets a bit blurry. The FRET gets a bit less. We don't almost see red anymore, but you can still see the quantum dots after 7 days. And you can also see that the survival rates are quite okay. So if you inject something, and you would also inject messenger RNA if you want to express the fluorescent proteins inside, if you inject something, there will be an effect, and you can see that the quantum dots we used almost have the same effect, so the survival rates are quite good. It's also important to measure the deformation rates because a lot of these lab animals survive. So often when you read the survival of the mice for cancer issues, the mice survived much longer. That just means that they killed them later, because usually they all survive. So they survive until the cancer explodes, because these are quite stable animals, so most of them are sacrificed. So deformation rates are also important. And there we can see, you see the old quantum dot. It's not that we didn't have new ones, but old ones try to aggregate, and then you can see that aggregates are very bad, so they can deform or kill these animals. There's other things you can do with quantum dots. I don't want to talk about this too much. That's, for example, we can build codes with these quantum dots to make, with this FRET, to make barcoding, single nanoparticle barcoding. We can use them for building molecular logic gates. So building some very low level computers, for example, with these systems. And we can also use other nanoparticles, as I said, upconversion nanoparticles to make these energy transfer, and also upconversion molecules to make this energy transfer for biosensing. And if you have any questions about this, I'll be very happy to talk about it. I would like to acknowledge some of the funding, our collaborators over the years, some people and companies, and the people who worked with me over the last 10, 15 years. And I'll end with these fluorescent images. And if you ever come to France, take care. They will see the FRET train, because FRET just means transport in French. But I was very happy when I came to France. I was in Germany before. So I make FRET over 10 nanometers, and these French guys thousands of kilometers from Lille to Marseille. So I thank you for your attention, and I hope I didn't stress the time too much. Sorry for that. Thank you. Finally, I remained on one place. Well, we tend to make them non-sensitive, and they're certainly not as these dyes that are, so they're pH-specific dyes, right, or environment-specific dyes. So there's no environment-specific quantum dots. There are some that bleach more or things, but I wouldn't use it for measuring a specific information. So because they could bleach because of anything. So what you would rather do there, if you were me, would put a pH-sensitive dye on the quantum dot and make the energy transfer from the quantum dot to the dye, because then you don't have dye bleaching because you excite the quantum dot and not the dye, but you still have the quenched or the lifetime sensing of the dye and can do that over hours or maybe over days, because your dye doesn't quench. Because that's the most problematic with the dyes, right? They bleach within a few minutes, and in that case they wouldn't. So this is also that has been done before, that fluorescein or other dyes that are sensitive to the environment are put on the quantum dots, and then you make the sensing by a FRET from the quantum dots. So you just use the quantum dot as the antenna to bring the energy to the dye.
Great talk.
Thank you.
I have a question about the quantum dots and the passivation. Because you show all these quantum dots as having a smooth surface and different things on there. But in reality, aren't there lots of organic molecules that are passivating the surface? And how do they just get out of the way of whatever you want to attach to the surface? Or do you have to worry about the presence of those molecules on the surface? And how do you get around that?
So that is the reason why when we use these his tags, we just have the organic molecules, and it's not the organic molecules that you synthesize them with, because then they're only in organic solvents. So you need to make an exchange of the ligands on the top, so to get them into solution. And the ligands we have that I showed for these his tag attachments, these are small molecules that stabilize the quantum dot nicely, so you can put them into pH 2 to I think 13. We've tried; they're quite stable. And the nice thing is that they're very thin, and they leave enough space for the histidine to come and actually coordinate them. Because we can also put a large polymer shell or PEG shell. So if you buy a quantum dot at Thermo Fisher, for example, they usually pegylate it. So it's a co-block polymer with PEG. They won't tell you exactly what it is, but it's quite thick. And so there we cannot access the zinc of the quantum dot. So in that case, we would need to functionalize the polymer on the surface and make the attachment to the polymer. Well, unless we use less polymer and leave it open, then they can still go inside. But it's a matter of density and of size and of how much do you need to make the quantum dots stable and water soluble. Absolutely. That's why we like the small ligands. So we like, well, we would like no ligands, but then they're not soluble. So we need some ligands to make them soluble and stable. But there we can access really nicely, because if we have 5 nanometers or 10 nanometers of polymer, it will be really stable. But we cannot make FRET anymore because we have this additional thick layer. I mean, 10 nanometers doesn't sound thick, but for FRET, it's very thick, so it's beyond the distance. So that depends on what you want to do with it. Yeah, but for FRET, we want a very thin and stable coating.
I was wondering about your antibody-based FRET assay. So for the antibodies, I'm just wondering how many donors and how many acceptor molecules do you conjugate to on their respective antibodies? Where on that antibody, what location on antibodies do you usually conjugate to?
So if we put the terbium or the dyes, as they're small, they're about a nanometer, and we would take a full IgG antibody, and we do labeling to amino groups, for example, and they're about, well, let's say 20 maybe on an antibody, maybe some more. So we can get 10, 15 on there. Usually we don't want too many, so usually we put 4, 5 to make the energy transfer, sometimes less. If we take the quantum dots, it's the inverse, because now the quantum dot is the big thing, and so we have several antibodies per quantum dot, not dyes per antibody, but antibodies per quantum dot. So for nanobodies, for example, we can put up to 30 on a quantum dot. For for full antibodies, we can put 3 to 4, and there we only have 1 acceptor then. But there is another advantage of these long lifetime, because if we have, so we made once a study where we had all these antibodies around, and we had 200 terbiums per 1 quantum dot. But if you have enough energy to excite all the terbiums, the quantum dot would grab the first exciton from 1 terbium and emit it immediately. But all the other terbiums will still be in the excited state, or at least 97%, because it's so long. So 1 quantum dot can de-excite hundreds of terbiums at the same time. If you have a dye, that would also be the case, but you cannot get so many around them. So it all depends on the size from the nanoparticle and from the dye. We can also label cysteine. So if we can introduce a cysteine, we did this with these nanobodies where we have 1 single cysteine. or with the alpha reps, that is nice because then we know exactly where our dye is. Because with the amino groups, it will be just all over. If we have a cysteine at a specific place, that would be nice for measuring distances. And in this case, we knew where it was, so we can measure the distance between two sites on the molecule.
So for the case where you have many ligands on the surface, can you use that population of ligands to look at dissociation rate, for example, where you correlate the emission to the event of unbinding?
Of the ligands? I think Ute Reschgenger in Berlin, she did a study about this, was I think in JAG some years ago, because it's true, people don't know how many ligands are on there. And I think she used that for making these, well, you can do a lot of complicated analysis, but as you say, if you could just look at the fluorescence and correlate this, and I think they made this with, I can't remember which ligand it was, to analyze the quantity of ligands that attach to the quantum dot by exactly that. I remember well. I will look it up and can send it to you. I mean, dyes are expensive, and quantum dots as well if you buy them off the shelf. So we buy a lot of quantum dots from Thermo Fisher. So they used to be Quantum Dot Corporation that was bought by Life Technologies, then Thermo Fisher, and then I think now, well, in the end, everything ends up at Thermo Fisher and PerkinElmer, right? So, but they are now under Thermo Fisher, and they are, if you buy them with streptavidine, for example, they're not cheap. I think they're like $1,000 for 2 microliters of I don't know how many nanomolar. So, but they have good ones. So they fired all their researchers about 10 years ago, I think, because it was too expensive, and they kept all their Russian synthesizers. So they make really good quantum dots, and the 625er quantum dots that we're using, they call them solar flare quantum dots. These are actually bought from our collaborators who have been doing quantum dots for 20 years, and they just put the ligands on it because they're so nice. So you can buy. There's some others that sell quantum dots as well. And I think now most of the patents of the quantum dots are beyond validity, so of 20 years or something. So I think there'll be more quantum dots now, but the standard ones are Thermo Fisher, so you can buy them.
Let's take, if there's anyone, or let me go back.